Hi everyone, my name is Jane Ogilvie and I'm a field ecologist at the Rocky Mountain Biological Laboratory. And I'm going to talk to you briefly today about my experience in doing long-term monitoring of bees, flowers, and their interactions. And so for some context, I established this long-term monitoring project because I'm broadly interested in bumblebee population ecology, and I wanted to better understand how environmental factors like floral resource availability and climate affect the timing and abundance of different bumblebee life stages during the growing season, and also how these factors affect overall population abundances. And so to get at these sorts of questions, I've been doing season-long monitoring of bees and flowers at sites around the Rocky Mountain Biological Lab for 8 years now. And so I visit each site weekly during the entire bumblebee active season. And during each weekly visit, I record the bees that I see and what flowers they're on during 1 hour observation periods. And at these same weekly intervals, me or a small team of very excellent people count the flowers that fall within belt transects that we have scattered throughout the sites. And so the first paper to come out of this long-term project actually just came out a couple of weeks ago with my collaborator, Paul Caradonna. So if you want a copy of it, please reach out to me. And so with this experience, I have three main insights to consider when it comes to designing monitoring projects. And the first is that quantifying floral resource abundance and interactions between bees and flowers is very informative. And so this is obvious, but flowers are food for bees. So if we have some measure of floral abundance, we have a greater insight into the presence or absence of bees or to bee population ups and downs. And monitoring interactions between bees and flowers specifically gives us details about what exactly bees are feeding on and how this can change over time. And it also gives us information about a bee's potential role as a pollinator in a plant community. And so it takes time to monitor flowers thoroughly, but I would say that collecting some sort of data consistently is definitely better than nothing. And so the second insight that I have is that the question should guide the monitoring design. And so we need to carefully consider the goals of the project because these goals will determine sampling frequently, what plants, species to monitor, how to count them. And so this can make sure that we're using our time efficiently. And so the third insight that I have is that natural history should guide our analyses. And so here I'm thinking specifically about when we're looking at relationships between bees and flowers. And so our selection of these variables should be informed by the biology of the system. So for example, the flowers should include the species visited by specific bees that we're studying, or we need to consider the time frame of flowers that most likely influence the bee variable that we're using. And so I found it really informative with this detailed knowledge about what bees are visiting to use species-specific floral lists when looking at relationships between bee and floral and abundance. And it's even more informative if we're able to scale these floral abundances by bee preference. So basically, flowers that are preferred by bees are weighted a little more heavily. And so that's it from me. I hope that this was useful. Thank you for listening. I hope you have a good day.